So, good evening, everyone. So, can everybody hear me? Am I audible? Okay.
Yes, ma'am.
So, we can begin our session. So, I'll be sharing the PPT. Okay. So, we are going to see about Blast. It has so much of data, but I have reduced it. I'll be only giving you the basic information because whatever research we are going to do, and at the end, the blast is the basic thing we will come and reach. So, this is Jita from Biowagon, and so we'll be providing many more classes like this on particularly on bioinformatics. So previously we had class on primer designing, and this is our second session. And I'll teach you as much as possible, like all the basic things, and if required, we'll be moving for advanced also. And 30 minutes will be of theory session, and remaining 30 will be practical session. And I'll be recording the session also, so we can move to the PPT. So, introduction. So, what is BLAST is a basic local alignment search tool. And why we use BLAST is like we have some sequences and we have to see what is the function of that sequence, and what what protein it codes for, and what are the species it is related to. So every basic things begins with a blast, and it is developed by NCBI. And NCBI is a gen bank, something which stores all the databases. And blast also helps in analyzing various kinds of research data. So we'll be seeing how we'll use blast in the functional, like in the further in the end of the PPT we can see how and where this blast can be used. So before going to blast, there is one thing we have to know, like what is this is a reading frames, because we will be using this reading frames in the types of blast. So to understand how this blast aligns, how this blast background algorithm works, so everything we have to know what is this reading frame. So we all know that three nucleotides code for one protein, and what the background algorithm of the blast works on two ways. Like it will, okay, I'll show you the cursor. So the forward will be segregated into three frames: frame 1, frame 2, and frame 3. Similarly, the reverse also, it will be segregated into frame 1, frame 2, frame 3. So totally we will be having 6 frames. So this 6 reading frames, to understand the 6 reading frames is very important. Like when you want to go, there are 5 types of blast. So when you have to analyze that 5 types, how it is working, like in which algorithm it is working, to understand all these things, this 6 reading frame is important. So it is nothing but, like in a frame 1, it will start reading the nucleotides, in with the first nucleotide. Here in case UCA, it begins from UCA, UGA. So this is a first reading frame, and it will code, it will translate to the protein which it codes for. In the frame 2, it begins from the second nucleotide, that is CAU. So it will translate to histamine. And similarly in the frame 3, it will begin with AUG, methionine. So in the similar way, it will also start reading the frames in the reverse order. So totally we'll have 6 frames for any nucleotide sequence you will be giving to the blast. So this is the basic thing. So why I have included this first, I'll be explaining you in the next slide. So we have 5 types of blast, that is blast N, blast P, blast X, T blast N, and T blast X. So the blast N is, we are giving the nucleotide sequences and we are searching against the nucleotide database. Similarly, blast p is, we will be giving the protein sequences and we will be searching again the protein database. So here the terms I'm mentioning as nucleotide base, protein base is everything. The in when you go to ncbi, when you go to the background databases of ncbi, you will have n number of databases. Like they'll segregate whatever data they have based on the uh, like if it is nucleotide, they'll keep it as nucleotide bases and protein database. Similarly, they'll also segregate the database based on the secondary database names. For example, you will be having pdb database, visprot database. So all such databases will be available in the ncbi. So here for nucleotide, we'll be blasting against the nucleotide, and for protein, we'll be blasting against the protein database. Similarly, for blastex, T-blastn and T-blastex, it is required when you're doing some functional characterization studies. So what blastx does is, so for example, if you're having some nucleotide sequences and you know that it codes for some protein, but you exactly don't know what it is. So in this case, that six reading frame will, it sorry it it will use the six reading frame method. So the nucleotide, whatever nucleotide sequences you are giving, first it will translate into six possible reading frame possibilities. Like it will have the six possibilities, and every possibilities will be blasted against the protein database, because here you know what protein it codes for. And t blast ns, it compares the nucleotide, compares translated nucleotide. So there is a huge difference between blastex and t-blastn. In blastex, you will be giving the nucleotide sequence as a query, but in t-blastn, you will be giving protein sequence as a query. So you may be confused now, like protein, nucleotide, all those things, but like when we are doing practically, we'll understand what it actually does. And I'll explain you clearly, and I'll be giving you some sample sequences of both the nucleotide and protein, and I'll explain you clearly in the upcoming session, like further. So here basically this is the difference between blastex and t-blastn. And what is t-blastex is, this is, we will use rarely, but it has huge function. Like when you are going for downstream studies, t-blastx is used mostly, but many people don't know that how helpful t-blast x is, because you can do the reverse way of blasting. Like you can go in a reverse order to find what actually the nucleotide sequence is in the t-blast x method. So here what we will do is, we'll give the nucleotide, and it will be translated into the six frames, six reading frames, and again it will be blasted against the nucleotide database. So here in this case, there will be almost 36 blast happening here. In, like for one sequence you are giving, it will be having the possibilities of 36 analysis, and you will get one unique report from the t-blastex. So t-blastex is a very effective way, and it can be used in the transcriptomic studies and all. Like we can see where this studies and how we can use all these types of blast and background algorithms. So blast runs with so much of background algorithms. So like when you go to the blast page and you search on the pdf they have provided, you can find how it is working, on what basis you are getting the data, everything. Here, the most importantly, it works on this four principles. One is seed and extended heuristic. So what it does is same like a primers. So if you give, like you'll give primers as a seed sequence, and you'll ask your PCR to amplify your target sequence. So similarly, what it does is it will take only the short sequences, and it will start matching with the databases it has in Ncbi. So whatever we are giving in a blast, it will match in a Ncbi database, because Ncbi is something which holds every databases in it. And a scoring system. So scoring system is, you will understand when you are analyzing the output, because it we have some like it works in the background. And I'll show where this scoring system, word based approach, and all these parameters where we have to give, and how we can modify the data based on our needs. I'll explain you in the practical session, so that it will be very helpful. But here I'm telling how it will, like basic things what are should be considered. And scoring matrices is a blossom and plum. Blossom is a black block substitution matrix, and a PAM is a point accepted mutation. These two are the major algorithm which works on the background. And like based on this algorithms, whatever I'm telling is, they'll have some biostatics things happening backside in the background of the blast. And we can change it, but mostly the parameters will remain same when you are giving blast, unless or until you need to have some other different research purpose. You can change these parameters, and otherwise everything will be like, mostly it will have a perfect accuracy of your result. And the most important thing in understanding blast is the terms similarity, identity, and homology. So what is similarity is like the likeliness, the degree of resemblance between the two proteins. So here you can also remember these terms, like when you are talking about similarity, you're talking about protein. And when you're talking about identity, it is about nucleotides. And when you're talking about homology, it is about gene. So I'm making it very easy because these three terms is very important when you're doing blast, or when you're reporting your blast result in the research paper. Because you can't write I have 80% identity of nucleotides, because identity is defined only for nucleotides, and similarity is for proteins, and homology is for gene. So now I'll tell how it is different. Okay. So if you are not getting anything of, or if you're having any doubts, you can send it in the chat box. Or you can't hear me, you can unmute and talk to me. So I'll replay everything in the chat finally. So what is similarity is likeliness between the two proteins. For example, if you are giving one protein sequence and you are getting the similarity of 80 percentage, means like these two protein is somewhat similar. Like that doesn't mean these two protein have same amino acid alignment. Amino acid alignments may differ, and here they'll use some terms called conservative substitutions. What is conservative substitution means, like amino acids are divided into various groups. They'll have a different structure, but they are biochemically same and their function is same. So that proteins are called as conservative substitutions. For, for example, we can take valine. So substitution for valine is isoleucine or leucine. In this case, when your query, so while I'm telling, mentioning query, that means the sequence you are giving. Whatever sequence I have doubt, I have like question, like what is this sequence, it is called as query. And what is the output it is going and matching, it is called as a subject. So like I'm giving force to certain words like query, subject, all these things, because it is necessary to understand what it is before going to interpreting the result. So that's why. So in the similarity, we'll be talking about protein, and I'll tell you how it is similar in the output. And in the identity, we'll be talking about nucleotides. It is either DNA or RNA. We'll be talking only about the nucleotide, that is exact match of nucleotide. If you are telling the identity is 95 percentage, means every nucleotide alignment in the two, in the query and subject, is exactly the same. So that is what it called as identity. And the third term is homology. What is homology is, it comes from the common ancestor. Like for example, the same gene between the human and chimpanzees. And it is also called as, over the time, their protein structure may be different, their nucleotide alignment may be different, but they come from the same common ancestor and they have the same function. So that is called as homology. So Currently, we will be using the term similarity and identity when we are analyzing the output. Homology comes only when you are going for taxonomy studies. So these three terms is very important when you are analyzing the blast. I'm repeating again, when you're talking about similarity, you're talking about protein. When you're talking about identity, you're talking about nucleotides. And when it is homology, you're talking about gene. So what are the parameters? Match, mismatch score, gap penalties, word size, e-value threshold, filtering option, matrix choice. So this is already most of the things is we already spoke. Match and mismatch is we have to, there will be certain scores aligned for match, mismatch and gap. Match will have a positive score, mismatch and gap will have the negative score. So based on that scoring we'll get the identity or similarity percentage. So there is one formula for that, I'll tell you that. And gap penalty is -1. And e value threshold, e value, what is e value threshold is expect value. That means when you are blasting something against the nucleotide database or protein database, you are expecting the uniqueness of your sequence. So it works in the opposite way. When your e value is low, that means your sequence is having so much of uniqueness, like it is significant and you can consider the data. And mostly we'll consider, like when we are taking the data from the blast, we'll consider e value as the most important parameter. So for some studies we will assign the e value, or basically the e value will be e power -5. That is 1 out of 1 lakh trials. For example, if you are giving one sequence and it is going through 100 of, like 1 lakh times it is blasting and it is giving one unique output, that is why we keep that eval threshold as e power -5. It is like when you're doing cancer mtt assays, you will have some significant p value is equal to 0.05. Similarly, in the blast, e value is very important. Lower the e value, higher the uniqueness and significant of the blast result. So we'll consider e value the most when we are looking for the from the output. And we'll also have the blossom and palm. We no need to go through in deep like what is blossom, what is palm, because it goes in a biostatics way. And it will be already fixed, this or parameters which will be constant towards all your blast. Only if according to your research purpose we need to change anything in this. And interpreting the blast output, so whatever the parameters you are giving, you have to interpret here also. Like you have to see what is the scores and location of matching, and e value, what is the accession number description. And you can also have a graphical representation. That means you can see the taxonomy and closely related species, everything you can interpret by blast output. So I'm making it short in a theoretical way because I can't explain you more in like in a demo session. And blast is also having two versions, that is web version which we will be using online version, and a standalone version is like a software. You can install it using a ubuntu or linux in your system, and it doesn't require any coding, but you have to know the basic linux commands. And we can come for the application purpose at the end of the session. So we can go for the demo session, and I'll be giving you the sequences in the chat box, so you can try it yourself with me and we can start analyzing the data. So till now, if you guys are having any doubt, you can message in the chat box. Sorry, I'm unable to share the nucleotide sequence because it is very lengthy, and we can go through. Like, I'll show you how to take the sequences from NCBI and how to do the blast. So I'll answer your questions at the end of the session, because when we are doing the output also you'll get so much of confusions. So can you guys see my windows? So you can sidewise make it a split screen and see what I am doing, and you can try it yourself. So first I have taken the protein T, that is P53. I'll show you, or I will send you the link. I have sent you the link in chat. Okay. So already during the primer designing session, I have explained how to look on the NCBI data. Like, okay. So this is the nucleotide sequence I'm taking, and I'm going to FASTA, and I'm copying this nucleotide sequence. So in the other section, I'm opening BLAST. So you can see the types of blast here, and we have taken the nucleotide sequence. So first we'll be trying nucleotide sequence. Okay. So whatever the additional parameters I have said, they have given in the algorithm parameter section. So you can see here what is the maximum target sequence. This is like number of hits, how much hits you require. So this will be standard 100 hits, and expect threshold. The 0.05 is what is that, 1 in 1 lakh possibilities. That's how it will be. And the word size, what the word size refers to, what I told, like a primer, the short seed sequences which it will be used. And match and mismatch score.
Excuse me, ma'am. Can you just repeat once? Like, I just missed it.
From where?
From last, like when you opened this server.
Okay. So first we'll go to blast. As I have taken nucleotide sequence, we are going for blast N. Here you can see the different types of blast. For example, in blast N, if you are giving the protein database, it will show you error because it works on a certain algorithm, and the input, you can see what is the input you should give here. Like in the blast N, nucleotide should be the input, blast P, protein should be the input, and whatever other things it will be constant. And only if you are in need of your research you have to change it. For example, if you want any organism particularly, you have to blast something against one particular organism, you have to give it here. And here comes the algorithm parameters. So these parameters are mostly it will be constant for everything. What is that maximum target sequences? When we get the blast output, I think, I don't know how many of you have noticed, we'll get 100 blast hits. That means it will give that first 100 blast hits as a output. So here we can change our numbers, like we can reduce it to like top 10 or top 50 blast hits, whatever it is needed. And expect threshold. So mostly the constant threshold we use is 0.05, that is 1 in 1 lakh possibilities. So it will give you the unique e value and lower e value among that 1 lakh blast output result. And word sizes, like I said, which will work as a primer. So it will take some 28 nucleotides and it will go and start finding it in the NR databases, and it will consider that okay, this is the sequence when you are doing blast. So mostly all these things will be constant, you no need to change anything. And here you can give what is the type of databases you want to blast against. Either nucleotide collection means it will blast against the entire nucleotide database. So you have a different types of databases here. You can also give genome database, RNA sequences. Only if you have to see particularly or RNA sequences, you can give it on RNA sequences, whole genome contigs, expressed sequence tag, or SRA files. You can blast against the genomic data, everything. So here overall we'll give NR database, that is nucleotide collection. And we have to give the sequence we have retrieved from Ncbi here. So mostly when you are giving FASTA format, this symbol is very important. Like sometimes only if you give this symbol and this accession number, it will find its sequences. You no need to give the entire nucleotide there. But when it is FASTA sequence, it should begin with this greater than symbol, because it is only then identified by the bioinformatics algorithms. Or else you can also upload the file and you can name it whatever you want, but all these things are not necessary. And if you want to have blast in the another result, give it in a new window and give blast. So whatever I explained in the first page is similar for all the types of blast. That is, for blast, NP, all this blast, the parameters are same, but output it will vary. Like you'll get different outputs. So I don't think whoever you have doing, you will get this kind of result page, because you will be having a different new version. This. So I hope you guys are doing with me. Okay. So when you are giving something for blast, some sequences for blast, this is the output page you get. And this page has everything in detail: what kind of program you have used, against which database you have given that blast, and what is the description, and what is the molecule type. If you are giving the protein sequences, it will show it as a protein. And query length, why I have told the use the words query subject is because that's how we are that's that part is required here when we are analyzing the data. So query length is the sequence which we have gave, that is 2512 base pairs. And you can see here show 100. So as I explained in the previous section, why it is 100 means we are giving only the first best 100 hits. So here we'll use the term called hits. That means the first uh, best 100 possible blast hits we are getting. And you can also change the requirements here, what are all the things you need. And you can see the e value is 0. That means lesser the e value, the more is the unique that sequence is. We have already taken this from ncbi, so it is showing that 100% it is matching. And this is the accession number, that means id number. So when we go through this, so whatever we saw is the description part. And next we'll have graphical summary. It will, like, you'll not have much data from this part. It will just show where are the differences. Like you can see the color here. If it is black, then it is having very less uh, less heat, so it will show the different alignments in this. And if we are going here, for every 100 hit you'll get this data. You'll get this one screen: name, sequence id, range, everything. So I'll explain you detailedly here. The sequence id is what which the id for the subject. uh, the We have given query, and it for which sequence in the nucleotide database it is matching. So that sequence id they have given here, and the length of it, and number of matches 1. I'll tell you what is this number of matches. Like I'll show you the difference and I'll tell you that. And you can see the e value is 0 and identities is 100%. So this is why I use the term identity, because we are talking here about nucleotides. So that means identity, it is having 100% identity, and every nucleotide is matching exactly the same. And gap, you are having zero gap out of 2512 base pairs, so gap is 0 percentage. And strand is very important, because what is that plus by plus is the numerator denotes to query and denominator denotes to subject. So it is exactly matching the forward strand or a positive strand. Like it is starting from 5 prime to 3 prime. And in some cases it will be in the opposite version. So you have to see against which strand it is your query is going and matching. So I'll show you the different types of result. Mostly when you go on to the lower level, you can find the differences. I'm searching for a plus by minus kind of a strand.
Ma'am, again, I couldn't get why it is plus plus only.
Like the numerator denotes what is the direction of the query, that is 5 prime to 3 prime. Okay, and the denominator denotes the subject's direction. That means both the strand which is blasting is in same direction. That is, when you are designing the primer, or when you have to know where the cds region starts, everything you have to know the direction. So that is why it is showing plus by plus. In some cases it will be plus by minus also. Hmm. Okay, so here it is showing plus by minus. That means the subject sequence is in reverse complement. So you can find that by these numbers also. For example, you can see here it is 1233, and it is going in a positive way, that is 5 prime to 3 prime, and you are getting the higher number here 1592. But when you see the subject, it is 7 7, that is 0848. But if you see here, it is going in a decreased order. That means the subject is in 3 prime to 5 prime direction. So you have to see which stand it is going and blasting. When you're designing primers, or when you're doing some indels identification, snp identification, you can't directly take the sequence which is showing in the blast result. You have to check all these sequences. And I also told what is this number of matches. So what is this number of matches is, there is something called hsp, which means when you are giving, 1 minute. Okay, so here the number of matches is very high, that is 1035. I'll show you with something smaller number so that it will be easy to understand. Okay, so in this case the number of matches is 2. What that means is, when you're giving some sequences, blast is having some algorithm called as hsp. What it does is, it will break the sequence. For example, this the query length is 2615. So if you are giving 2615 nucleotides, it will not blast entirely 2615. It will break the 2615 into smaller segments. For example, 200, 200, 200, and it will try to match exactly that 200 sequences same in the subject also. So here you have to be very clear what I'm referring to as subject and what I'm referring to as query. Query is the sequence which we are giving. Subject is the ncbi sequence. Okay, so here you can also change the value based on query start position. Okay, so as I said, this 2615 sequences will be break into many seed segments, and it will be blasting. So in this case, it is exactly showing that from 1 to 114, whatever the query we have given is matching exactly the same in the subject also. But from 114 to 173, there is a huge gap. So if you see the subject position, uh, between like almost 60 to 70 nucleotide is missing in the subject. Here it is ending with 114, but here it is starting with 217. That means there is something other nucleotides in between this section. So whatever the query you are giving is exactly not same. In in this case you have to go to this gene sequence id and identify what is this additional sequence. And according to your research, like if you are doing something in snps or indels, you have to know there is any possibilities of insertion or deletion in that missing sequences in the query. So that is why we have to focus on number of matches, because when we when we see the number of match is one, that means your alignment, your blast alignment is significant, and you can consider that for further process. But when you are seeing the number of matches is 2 3 or more than that, there is more possibilities of mistakes, like missing nucleotide sequences, or there may be additional nucleotide sequences in your query. So you have to find what is that uh, missing thing. So in this case you have to go to the original subject sequence and you have to see what are all the nucleotides present from 115 to 216, because that may be important or may not be important. But you have to know, like when you are analyzing the output, you have to know what are all the things you have to consider. Here you can also sort it in a different positions, like you can also keep it like a subject start position, query start position, or percent identity. So you can make it however you are convenient. So I'm telling you how to analyze the output and what are all the things you need to consider when you're seeing the output. So I'm not going in detail like what is that missing sequence, what will be the things there. So I'm not going such things. I'm just telling when you're getting the blast output, what are all the things you have to focus. Okay, this is for nucleotide. Similarly, I have given you the protein sequence in the chat box. So I'm closing this nucleotide and I'm going for blast P, and I'm providing the nucleotide database. So if in blast N I'm providing the protein database, and if you give blast, you'll get this error like protein FASTA provided for nucleotide sequence. That means in blast N you should provide only the nucleotide data. So that is the thing you have to consider. And I'm like the other parameters, whatever I told for nucleotide, it will be the same here. So it is taking time. So I'll be giving, we can give the input for all the blast X, T blast N, and T blast X also, so that it will be easier. It will be running in the background. And in the T blast X, you have to provide the nucleotide sequence. Clear. and here you have to provide. Okay, so we have given the protein database of the same TP53 protein. So it will have the same kind of output, everything, and alignments. So when you are seeing the protein protein match, also you'll have the same number of matches. Here you'll not have the direction, because when you are giving the protein sequence, it ultimately begins from start codon, and it will have only the 5 prime to 3 prime direction. In this case, you will not have the signs whether it is a positive or negative strand. Only when it is nucleotide, you will be having that plus by minus kind of thing, and you will have number of matches here. So there is something in protein output which we have to focus, that is between you will have some gaps. W- Here you can see some gap here, and similarly you will have something like positive symbol, or something you'll be having an so what does this means is L and X, the protein L and X is not, it is not like a substitution protein also. These two are different proteins coming here, so we call this as a space. So all this space, gap, match, mismatch, everything will have some scores which the matrix will align, and it will give it in identity. You can see the space here. Hmm. So here you can see plus. So what is plus is positive match. That means the protein R and Q, they can be used as a conservative substitution, because the two proteins comes under same family. They have same biochemical function. So it doesn't differ that much when it is coming ultimately when it comes to protein function. When this nucleotide will not have any effect in that protein, but if you are having space, you consider that it may be a deletion or insertion, some indels may be possible. So that is the only difference between the protein and a nucleotide database. In protein database, you will have such things you have to focus on, and rest similarly you'll have number of matches also. You will be having um, different more possibilities also you can find. So you have to focus here also. And okay, the alignment view. So this will be pairwise alignment, that is constant for the blast output. This algorithms, like whatever I'm telling is blossom, palm, pairwise, and global alignment, local alignment. So all these things you need to have a separate session, because it all needs a biostatics, and it requires some time because it involves so much of mathematics in it. So that's why I'm not going very deep in the matrix and all those algorithms. I'm just telling whatever output you are getting in blast, and what are the important things to focus when you have to consider for your research purpose. So there is nothing difference in nucleotide and protein. The only difference is the positive space whatever you get in the protein sequences. And when you go to blastex, you have to provide the nucleotide sequence. So as previously I said, blastex, when you are giving a nucleotide sequence, it will translate that nucleotide into 6 possible ways. So I'm repeating it again and again, because you have to understand the difference between only blastine, only blastex and T blastex. So you have to understand the differences between the each blast. So that's why I'm repeating this again. So in blastex, the nucleotide which you have gave will be translated into 6 possible reading frames, and each reading frame will be blasted against the protein database. So I'll provide you the PowerPoint. Also, I have made additional PDF also, which contains minute details of whatever I'm telling now with a simple examples. I have made PDF also. I'll try to upload that also. So you guys also can try in the next tab for all the requirements. So if you're having so much and if you're confused, please check here what is the function you are running. Either it is a blast text or blast, what your program you're going through. So I'm closing this blastex. Okay. So while analyze, okay. The additional thing you have to focus, like you have seen something different in blast n. That is, you'll be having a strand plus one, that is plus or minus strand. In the protein sequence, you will not have anything such as strand, like which direction it is going. In protein, you have to focus only on gap, mismatch, everything. So the thing which is different in blastex is frame plus 2 frame. So as I said in a theory part, you will have 6 frames. That is, your nucleotide will be going through 6 frames. In that, it is telling that second frame is having the high identity. So you have to see which frame it is matching here. So you can also confirm this by giving the same sequence in ORF finder. So when you are giving something in blastex, sideways you can keep one tab open, like open reading frame bioinformatics. So there is one online server for ORF finder. So in that case, you have to give the same nucleotide sequence there, and you have to see what is the ORF length, and which strand it is, whether it is in a positive strand or negative strand. All these things you have to find in another website. So this is something different from the previous things which we have noticed. Here also you'll get so much of things like number of matches, everything. And the when you're seeing the blastex output, the one thing you find which is different is isoforms, because your nucleotide will be translated into 6 possibilities, so each possibilities may be a different isoform. So when you're giving blastex, you're having possibilities of finding so much of isoforms here, and that is what it makes unique from blast n and blast p. Okay. So if I'm not telling some of the things, that means it is similar in all the three programs. So here I'm giving T blasten, that is the nucleotide. Will be giving the translated nucleotide. Sorry. Yeah, it will be searching against the protein database. No. So like when we are going through all these programs at once, we'll be having so much of confusion where to use blastex, where to use T blastex, where to use T blast and everything. But when we are constantly analyzing the output, it will be very easy. Like under which condition we have to go for T blast X, under which research application we have to go for T blast N, so we can understand where we have to use what kind of program. But before that, we have to understand how it is working. That is the basic thing. Like what is the input you are giving, and what is the output you are getting, and how the output is generated, and you have to know what is the different thing you have to focus on that output. Like we saw three programs, and the three program has some uniqueness. When you are seeing nucleotide, when you are seeing protein, when you are seeing translated protein, you have a unique output. That is something different which you have to focus. So what here I'm telling is, you have to focus on the minute details in the blast output, because you can't design primers in the 3 prime to 5 prime direction. Like you can't make it as a forward. So you have to know all these things. When like it depends on what research outcome you are expecting, like why you are using blast. So what it all depends on why you are using blast, for what purpose you are using blast. So for that, you have to understand the basic. And that's why I thought of starting with the blast. Like this is very basic, and I thought of including all the algorithms, everything. But it is 8 o'clock, 1 hour, and I couldn't complete it within 1 hour. So for a like advanced blast, it may take some more time, like 30 minutes more or less. Okay, so what is the function we have given is t blast in. That is, you are giving a molecule type, you are giving the protein database, and you are asking it to search or blast against the nucleotide database. That means like 6 reading frames. So you can see the output of T blast 10 and blast X is same, almost same. What is the only difference is, it is searching against the nucleotide database. So in BlastX, only the query will go for 6 frame reading section. But in TBlast10, both the query and subject will undergo 6 reading frames, and it will blast against the protein database. So you can see there is no difference between the previous output and this output, because whatever we saw is almost same here. Like you are having frames and matches, everything is same here. And we can see in T blastics. So Aman, look, until it is loading, I'll try to answer your chat questions. So Aman has asked the term homology and other is like a central dogma. So in bioinformatics, like normally in a science, what you consider as a central dogma is DNA to RNA, RNA to protein. So in bioinformatics, what you consider is sequence to structure, structure to function. So sequence, like you will go for nucleotide, same similar DNA, and structure, that is protein, and function. So in the central dogma of bioinformatics, like if you consider, it will be sequence, structure, and function. And the term homology, I didn't actually understand what is the difference between term homology and central dogma you're asking. Ma'am, what is the background algorithm consist of and how it can be built? So if you want to build background algorithms, you have to know the matrix. Like it will be very easy actually when you understand, because only you have 4 nucleotides, and 4 nucleotides you will be having only 4 numbers, but you have to know how you're giving that numbers. Just that is the only difference. If you type blossom, pam, pairwise alignment. In Google, you'll see some matrix structure with a -2, -1, a huge, almost 9 to 9 cube box you'll get. So you have to understand what it is. So it will take another session. Actually, it is very, because you have to do so much of maths. Before going to matrix, you have to understand what is that alignment: pairwise alignment, local alignment, global alignment. So you have to understand all those terms. What is positives and identity? So, okay, what is positives? A positive, 1 minute, control F. Okay, for example, if there is positive here, so I'm marking it as D, but you imagine there is positive there. That means that the amino acid in your query and the amino acid in your subject belongs to the same family. And they both are maybe different structurally, they may be different, but they have same biochemical function. So even though there is that substitution, the ultimate function of that protein will not differ, and it will be same. There will be no effect in the function. So that is why it is called as positive. So the, and identity, as I said, what is identities? Identities refers to the nucleotides. This is T blast end. That means whatever the nucleotides you have, it will undergo 6 reading frames, and it is giving the identity here. Okay, frame 2 and frame 3. 1 minute. So if we see here, whatever the sequence we give, which will be translated into 6 reading frame, it will be translated in this case frame 1, frame 2, frame 3. This is for forward. Similarly, we'll have frame 4, 5, 6 for the reverse complement strand also. Here it is telling that it is starting from the second nucleotide. It will not differ that much, like we have to know this data, but we have, like when we are designing primers, we'll give the same nucleotide in the ORF region. 1 minute. I'll show that. Okay, so I think you can understand here what is that a frame 3, frame 2, everything. For example, I have taken some nucleotide sequence, and I have to design the primers, and I have given the same nucleotide sequence in the ORF finder also. So you can see here the frame section, that is positive positive 2, that is means 5 prime to 3 prime, second frame it is working. So here ORF1 is, it is starting from nucleotide 1, ORF2 is, it is starting from nucleotide 2. So this will be easy. It's like cross confirmation. You are getting the same frame in ORF and in blast results also, so that you will not have any mistakes in your primer designing. So that's why I have told, like we have to focus on that frame section. And okay, here in T blast 10, we'll find no significant similarity found, because when we go to T blast X, there is a possibilities of you have okay, now consider you have taken the same nucleotide sequence, same protein sequence for all the five types of programs, but in each program you are getting different outputs. In T blastex you didn't find any similarities, but the function of this T blastex is something different, because it is searching, it is translating, like it is breaking your frames into six, and it is also breaking the database frames also into six reading frames, and it is blasting against the nucleotide database. So where you use this particular T blast axis when you are doing genomic or transcriptomics analysis, you can use this T blast X. For example, you are having a huge data, it's not something from Ncbi, but something the data you have generated in your research, you don't know what it is. So in that case you can do T blast X and you can find where the nucleotide sequence is. So you can see that T blast X will not be represented in the first page also, because it is used very rarely, and it depends on what type of research you are using. But other four blast programs, these four will be used constantly throughout many, like many kind of applications are there. And now I'm not going to show primer designing. Actually we already had the primer designing session, and we are also 10 minutes ahead of our session. So if you guys are having any questions you can ask, or else if you have any doubt in our session whatever today we thought.
Ma'am, can you please give me the definition of blast as I joined late? So,
So I'll be uploading, yeah, I'll cover the application part. So definition of the blast is it is a basic local alignment search tool. When you have some sequences you don't know what it is, you'll find the sequence, function, name, everything from the already available databases. So that is the blast. And I'll upload the session, you can go through like from the beginning, because it's already taking time. I'll cover the application part.
okay.
So like as I said previously, blast depends on for what kind of research you are using. You can I have I have named some of the applications, but actually when you are doing some research, and when you are doing like big research, when you are doing transcriptome, when you are doing Sanger sequencing, whatever it is, but ultimately will come and end with a blast. So it is the basic things, and it is the easiest thing to learn when you want to learn something from the bioinformatics, because we'll be starting more bioinformatic classes, so that's why I started with the blast. So you can go for homology search, like identify the similar protein sequences. I'm not going to cover entirely what all these things, because it can be used everywhere for drug purposing, metagenomics, genomic studies to find the protein structure. Actually, when you go for something like a protein blast, you can also design homology modeling based on the identity, all those scores. So we'll have that separate session on docking, homology modeling, everything. So I'll explain there clearly how this blast will be used there. And blast, why blast? There are many other tools which does same function like blast, but every each of the function has pros and cons. If you go for blast plus, you have to use it in a Linux command, so it may be hard for your beginners. So everything has only limited. If you go for ensemble or something, you will have access to only limited databases. But when you are doing blast, you will be accessible to ncbi databases. So whatever the primary secondary databases they have submitted their data in ncbi, you will have access to it. So whatever the output you are getting, it will be ultimately more significant when compared to other databases. But we can also use other, because every database has its own pros and cons, and what it is and how it is differed from ncbi blast. I have included everything in the pdf, so I'll make it available. So blast is not only the tool for alignment or for doing all the basic uh, se- like sequence analysis. There are many tools like a blast, but significantly it is very helpful, and the output we get from the blast is from the major source, that is ncbi. So that is the only difference.
So basically, blast is used to analyze the sequences.
Yeah.
Okay.
Okay. In future, are you going to talk on primer designing again? So I don't, I can't exactly tell we'll be having a primer designing class or not. But if like many people wants to have the class, we'll think of it like conducting it again.
Ma'am, as you tell what is difference between protein and nucleotide blast, but I can't get it your point.
Like what, in which way you are asking?
Protein blast applications, matlab, I can get the nucleotide blast, but I can't get your point at the protein blast.
Nuclear protein blast is similar to nucleotide blast. Like in protein blast, you'll give nucleotide. Sorry, in nucleotide blast, you'll give nucleotide as input, and it will
Okay,
search against the same nucleotide database.
okay, okay,
Similarly, in protein, you'll give protein as an input, and it will be searched in protein database.
okay.
So there is no
So,
much difference. But when you are getting output, I told what are the things you have to notice in the output section.
okay. In protein blast, what is the meaning of plus sign? Is a positive? What is positive for protein blast?
Yeah, the positive sign denotes the substitution proteins. That is, the proteins which have different structure but same function.
Okay,
So
ma'am.
that means your ultimate protein function will not differ by the substitutional proteins.
Okay.
So already we are ahead 15 minutes. So if you guys have any questions also, you can ask in the WhatsApp group. And um, I hope you all had something, learned something new about blast today. And I'm expecting the feedback if something uh, I have to change, or something should be included, or something should be neglected, whatever the feedback from this session. So you can give your feedbacks in the WhatsApp group, and we'll work on it from the upcoming sessions. And also you can give us what are the topics you are expecting, like bioinformatics topic you are expecting us to take the classes. And we'll also have something like one-on-one session. Like already I have been conducting this one-on session with a few of my students. Like if they are having particularly doubt in their own research project, we'll be helping that person on like we'll be guiding whatever we know. So such sessions also we are conducting, and already 3 to 4 people we are helping. So if you guys want any help there also, we can help. So whatever you feel, whatever you want to learn, you can easily, like you can share it in the WhatsApp and we'll reply you immediately. And I hope uh, this session was something informative to you guys, and I am expecting feedback from most of you, like in the WhatsApp group, so that it will boost us for our next upcoming sessions. So thanks for your patience. Like it's already 15 minutes ahead of our session. So thanks for attending everyone. And so you can leave the meet, but don't forget to give your feedbacks in the WhatsApp. Thank you.